here introduce Dr. Emmanuel Antonarakis. Dr. Antonarakis is a Clark Indoe Professor in Medicine, and he's also the director of our GU program. He plays a crucial role in the Cancer Center as an associate director for translational research. I was told I should keep this brave. And so, looking at his CV, the awards and achievement he's done, it will take this whole seminar to talk about that. But as a friend, a collaborator, when I think about Emmanuel, I thought about an action-driven problem solver who really have this very deep understanding of biology. But most importantly, what really touched me is that he's always worked tirelessly to translate research finding to best therapeutic choice for patients. So here, Dr. Antonarakis.
Thank you, Stephanie, for that kind introduction. And I want to talk today about something.
Sorry, we've got some noise coming through the mics that they.
Okay.
Chiming. So yeah.
I don't hear it now, Joel. Sorry for the disruption.
We'll try again. Good morning, everyone. Thank you, Stephanie, for that lovely introduction. And I value our collaborations and friendship very much as well. I'm going to talk about one of our high-risk, high-reward projects. And if there's any trainees in the room, whenever you do a project that involves risk, you should be prepared that 90% of the time it's not going to succeed. And I had about 10 of these projects that I was doing simultaneously, and the other 9 failed. And I don't want to give a lecture about the 9 that failed, but about the one that succeeded. But risk does sometimes come at a cost. And these are my disclosures for this talk. The high-dose testosterone that we use for these studies is generic. There's no commercial sponsors, so none of these studies is influenced by any pharmaceutical companies, but those are listed there. And I like to start my talks about prostate cancer talking about the so-called clinical states. Unlike other cancers where we become obsessed with the TNM classification, prostate cancer, we actually don't ever talk about TNM, but we do talk about these clinical states, starting on the left-hand side with the localized curable disease, moving on to the biochemical recurrence, where patients have a rising PSA after definitive local therapy, but no radiographic evidence of metastases. Moving all the way to the far right, where these patients have metastatic disease that has become refractory to hormone deprivation therapies. All the gray boxes show the FDA-approved therapies and their location in the treatment sequence. And you can see a lot of different classes of agents there, but just to make your life a little bit easier, all the ones in red are the hormone-related therapies. These are treatments that either block testosterone production or the testosterone from binding to its receptor. And the field of prostate cancer has been dominated by these androgen deprivation therapies or anti-androgen therapies. And by the way, if you look at the far right, and you'll see some other drugs in there, including immunotherapies and chemotherapies, please remember that these patients, when they transition to those other treatments, they continue their lifelong androgen deprivation therapy. So it's androgen deprivation plus one of those new things. So really, hormone blockade has been the cornerstone of prostate cancer for a long time. Why is that? Let's start with some history. So we've had the fortune of 2 Nobel Prizes awarded to prostate cancer researchers. Charles Huggins, a urologist, he's actually Canadian, but worked most of his adult life at the University of Chicago, and an endocrinologist who is Polish, working at various locations, including Bayer and Tulane. In 1966, Charles Huggins discovered that when he performed a surgical castration of a man with prostate cancer, his tumors melted away, and he was so surprised and shocked, and he used to keep a diary, and he wrote in his diary, mid-1940s. He said, "I think I found the cure for metastatic prostate cancer." Can you imagine his excitement at the time? And then the future diary entries started to say, "We've seen the first relapse, we've seen the second relapse." And he then went a step further, and he had realized that not only do the testes produce testosterone, but the adrenal glands. So his second phase of research, he started to do adrenalectomies, surgical removal of the adrenal glands, and he could salvage some patients who subsequently had further disease control. Andrew Shalley's main discovery was he discovered the hormones that govern androgen production: luteinizing hormone, releasing hormone, follicle stimulating hormone, and luteinizing hormone. And 5 years after his Nobel Prize, he discovered the first drug that agonizes LHRH and subsequently decreases testicular testosterone. So the first medical therapy for testosterone lowering effects. And since that time, the field has been obsessed, including my own research, with annihilating or extinguishing the androgen receptor in any way possible. This has really been the central dogma. So the androgen receptor is a steroid receptor. It's inside the cytoplasm. It is bound to heat shock proteins in the cytoplasm when it's not bound by its ligand, which is dihydrotestosterone or testosterone. When testosterone binds to the androgen receptor, it moves from the cytoplasm, dissociates from the heat shock proteins, enters into the nucleus, dimerizes with itself, forming a homodimer, and then binds to the enhancer or promoter regions of a number of genes, including the PSA gene itself, which is why PSA is such a great measure of androgen activity. And all those blue squares show therapeutic ways that we have tried to annihilate or extinguish AR signaling from the first node to the last node. And these involve inhibiting the adrenal enzyme that produces testosterone, inhibiting the androgen receptor itself by potently binding to the ligand binding domain, binding to the N-terminal domain, which is the transcriptional activation domain, degrading the androgen receptor through proteolytic chimeras and other ways, blocking CYP11, which is the first enzyme in the adrenal gland that converts cholesterol to pregnenolone, and then other epigenetic regulation where we are inhibiting the co-transcription factors and pioneering factors that enable the androgen receptor to maintain its transcriptional control. But now let's go to a definition. So what does iconoclastic mean, or the iconoclastic movement? It comes from two beautiful Greek words: icon, image or figure, and clastic, to break or destroy. You've probably heard of the osteoclasts, which are the bone-breaking cells. So historically, in the early Christian church, this was the social belief of the importance of destructions of icons or images or monuments, most frequently for religious and political reasons. And on the right side, you can see a statue of saints, and the heads of the saints have been bashed away. In modern day, this is used to refer to attacking or opposing generally accepted beliefs, dogmas, or traditions. So today I'm going to tell you a story about iconoclasm, where we used the 70-year paradigm of androgen-directed therapy to flip it on its head and to do the exact opposite, which is to stimulate the androgen receptor rather than inhibit it with supraphysiological high doses of testosterone. And in my days at Johns Hopkins, my office was right between the offices of Sam Denmead and John Isaacs, two laboratory researchers who made the preclinical observations, pioneered this approach. And as they were talking outside my office, I was overhearing some of their conversations. And after a while, they came into my office and said, "Why don't we design a clinical trial to explore the preclinical data?" And the preclinical data showed that in several prostate cancer cell lines, but not all, for example, the LNCAP cell line and the VCAP cell line, giving back supraphysiologic doses of androgen, in this case R1881, a synthetic androgen, caused more growth inhibition of those cells than bicalutamide, which at the time was our most potent anti-androgen. This was not the case in all cell lines. For example, LAPC4, this did not occur, and with LAPC4, if you gave androgens, you actually stimulated the growth of that cell. And on the right-hand side, this is a LINCAP model where they show that the high-dose testosterone initially causes cell killing, but over time, the cell adapts and then begins to utilize that high-dose testosterone to begin growing again. And Sam and John in their laboratories had postulated a number of mechanisms by which this might work, and I'll explore some of these with some of the clinical data that came. So how did we decide to do this in humans? We coined the term bipolar androgen therapy, not because we made patients psychiatrically ill, but because we were alternating between extreme poles of testosterone concentrations. Remember, these were all men that had been androgen suppressed for a long time. They had now become castration resistant. Their testosterone was low, and despite that, their cancer was progressing. And then what we decided to do was to give these patients an intramuscular, supraphysiological dose of testosterone, which rendered their blood testosterone levels at least 1,500 nanograms per deciliter or higher, which is about 3 to 5 times higher than physiological levels. And the paradox was that we maintained these patients at the same time on their medical androgen deprivation therapies. And we did that because we wanted to ensure that after each of these bursts of high-dose testosterone over the course of the next 28 days, 4 weeks, their testosterone would then return back to the castrate or near castrate range. And 28 days later, we picked the 4-week interval for pragmatic reasons. We gave them another dose of high-dose testosterone. When I was 3 years into my faculty job at 2013, I met Michael Schweitzer, who was one of my first fellows that I mentored. And Michael was hungry for projects. And I said to him, Michael, would you like to design the first trial looking at supraphysiologic doses of testosterone in men with castration-resistant prostate cancer? And he took the bait. And in this first trial, we were really believers in the fact that one of the ways that bipolar androgen therapy or supraphysiologic testosterone worked was by inducing double-strand DNA breaks, perhaps similarly to a platinum chemotherapy or even in modern day PARP inhibition, for example. So we wanted to prevent the repair of those double-strand DNA breaks. So we used a high-dose testosterone in combination with etoposide. This was the first and last time that etoposide was part of the treatment, and after this we abandoned it. But in my 17-year career of doing clinical trials, this was the first and only study that was ever disapproved by the IRB. Not tabled, not minor stipulations, not major, disapproved. And we showed all this preclinical data, and we went out of our way to show how in the cell lines and the xenograft mouse models this worked, and the IRB said, absolutely not. This is going to hurt patients. It might even kill some patients. So as we always do when we get a disapproval, we started to bargain with them. And we said, what if you allow us to treat 4 patients at a time? If at least 2 of the 4 have a favorable response and no toxicity, we'll treat 4 more, et cetera, and we'll get up to 16. And after 16, we'll stop and reassess. So under those very strict conditions and a lot of safety rules that were built into the trial, they allowed us to do that. And this was the first study which we had an acronym for it. We called it the LOVE study, LOVE, because we thought that patients would be able to make love again after all these years of having no sexual function. And this graph shows patient 001, our very first patient. And this is a man who initially scared us because his PSA, as you can see, went up with the first 2 to 3 doses, but he didn't have any clinical symptoms, and we continued it. And then by the 4th, 5th, 6th, and 13 cycles later, he developed a remission. He also had a very large, as you can see on the right-hand side, pelvic sidewall lymph node, but pretty enormous actually, which shrunk. So we got through our first 16 patients, and 8 of the 16 had a biochemical or objective response. Unfortunately, one of the patients developed neutropenic fever from the etoposide and actually passed away of sepsis that we could not treat. So we had a death rate of 1 out of 16. So that almost killed the future development of these drugs. But we were able to argue that this was not from the bat, but was from the etoposide. So over the last 10 years, between 2014 and 2024, We've conducted 8 trials, and we've treated over 400 patients. I'm going to fast forward now to the last 2, which I think provide the deepest biological insights, because we finally began to explore biomarkers after realizing that we didn't really know the cause of these responses. But before I get into some of the biological insights, I just want to give you a snapshot of what we've learned over the last 10 years from a clinical perspective. First question is, when is the ideal time to give BAT? And we believe that the ideal time is after a patient has received androgen deprivation therapies, the disease becomes dormant for several number of years, and then, as we often see in the clinic, their PSA begins to rise, but they're not having any clinical symptoms. Their scans appear stable, and the only sign of progression is rising PSA despite castration levels of testosterone. And we think in this setting, where the potential risk of harm is very, very low, and the risk of benefit outweighs the risk of harm, this is the ideal scenario to use this treatment in, and of course, typically prior to chemotherapy. Over the course of about 400 patients, we have seen 3 broad responses. About a third of patients are depicted in the red lines, where we see a complete or partial PSA response and radiographic response. In about a third of patients, we see a stabilization of their progression, so the PSA in their scans stop getting worse, but they also do not get better or regress. And then in about a third of patients, the disease either continues at the same pace or in some cases appears to accelerate a little bit. So that's important clinically, because this should not be done outside of centers that have experience with it, because that last group, the orange group, where you might have progression or hyperprogression, can be scary. And then to further add to the complexity, we have seen this concept of bone scan flare. Chuck Ryan will appreciate this because he reported that happening with abiraterone. So look at this patient here, baseline PSA 124, he gets 3 doses of high-dose testosterone. If you were the radiologist looking at that second bone scan, you would be freaking out and you would say the disease has progressed. We had the clinical knowledge that his PSA dropped from 124 to 14, and the fact that this patient had no new symptoms. In fact, his bone pain was improving. So we kept him on the trial despite the fact that our radiologist said this is progression. And he was on study for more than 12 months, and his PSA continued to drop. And by month 12, his PSA was 0.4. So this highlights some of the challenges of interpreting whether this thing is even working or not, because you could have these artifacts. And of course, we've had other patients where their bone scan looked like that on the second progression and their pain was worse. So it's really hard to disambiguate sometimes the clinical progression from pseudo-progression. After publishing about 3 or 4 of these papers, my email inbox began to be flooded by patients and physicians from all over the country and the world asking me advice about how to safely give bipolar androgen therapy. And I was taking an enormous amount of time responding to each and every person. So we decided to circumvent that by writing an article that was specifically geared towards patients and physicians, not to oncologists, not to our peers. And this paper was very, very hard to publish because no journal wants to publish a patient's guide. So we ended up, and now when I get those emails, I just attach this paper and I say, do what it says in this paper. We have a number of pearls in there and cautionary notes. Now, Dr. Denmead, shown on the top right, and I designed a very interesting study, which we called the Transformer Trial. This was called the Transformer Trial because it was funded entirely by a DoD Transformative Impact Grant. They give out one every year. It's $5 million. And we funded a 200-patient study purely with a DoD grant with no industry sponsorship, which I was quite proud of. And this was a nightmare to do because we did that at 18 sites, and Johns Hopkins was a CRO because we didn't have enough money to hire a CRO, although we took out a lot of extra paperwork. And this was a study where we took patients at that era, they were getting ADT, and then they were getting abiraterone. And the abiraterone was either given concurrently with the androgen deprivation or sequentially. And these were patients that had not yet received chemotherapy. And we randomized men equally to receive enzalutamide, which at the time was the most potent anti-androgen that we had, or the exact opposite, the most potent androgen receptor agonist that we had, which was high-dose testosterone. And we used radiographic progression-free survival as the primary endpoint, but we decided to build in a crossover such that patients who were randomized to the enzalutamide, if they progressed on their scans, they were then allowed to get the bipolar androgen therapy, and vice versa. So that the carrot for patients was, if you're randomized to this trial, if you stick with it long enough, you're going to eventually get the bipolar androgen therapy, because they were all excited about getting that. So the first thing that we found was, and this kind of blows your mind because you're using two diametrically opposing therapies, your strongest AR antagonist and your strongest AR agonist. And when you do that, you see very, very equivalent results. The blue bars show the PSA responses to the bipolar androgen therapy, 28% met the 50% threshold. And on the right in red, 25%. These were not statistically different, but at least we could say that they were numerically equivalent. Now, we didn't build in biopsies in this first study, but we had one patient who allowed us to perform a baseline metastatic lymph node biopsy, and then a second biopsy after 3 cycles, in other words, 3 months of bipolar androgen therapy. Everybody at Johns Hopkins at the time was debating as to whether at the protein level the androgen receptor would go up in these samples or whether it would go down. And people had their own arguments about both of those. And what we showed here for the very first time, and I still remember our pathologist calling me on the phone, and he was so excited. It was Angelo DiMarzo, in case you guys know who that is. He said the androgen receptor protein is not only diminishing in a big way, it's hard to see here, but the localization of the AR also becomes more nuclear. So on the right-hand side, even though you see less brown staining, you can see the nuclei coming out more, and there's less AR in the cytoplasm. Where on the left side, you can't distinguish the cytoplasm from the nucleus because the AR, which is not bound by its ligand, is in both compartments, the nuclear and the cytoplasmic. So this was
the same size?
The node was about 80% smaller. And as you can see, there's also less tumor in the node. So that's one of the biases why there's less brown. So hold that thought for now. We designed this study with the naive and ambitious expectation that the bipolar androgen therapy would be superior to enzalutamide and superior in terms of survival as well. So when we submitted this paper to the New England Journal of Medicine, it was reviewed, and all 4 reviewers had very positive comments. And then the editor-in-chief said, I'm forced to reject this. And he said, the reason I'm going to reject it is that you do not meet your primary endpoint of superiority. This was not designed as a non-inferiority study. So we took those comments, addressed them, submitted that to the JCO, and it was accepted in less than 1 week. But the point is, we did not meet our primary endpoint. And as you can see here on the left, radiographic progression-free survival, no different. Even if you can't read the p-value, it doesn't matter. And on the right-hand side, overall survival, no different. But we were very interested in what happened at the time of crossover, and this was really enlightening. And this was our first study that we had a crossover, and I'll show you some of those data. Now, if you look at the forest plots for progression-free survival, there was one category here where the benefit was different in one group versus the other, and that was in patients who had become resistant to their prior abiraterone in less than 6 months, did better with high-dose testosterone, and those that had been on abiraterone for longer than 6 months, in other words, they were more hormone sensitive, those patients did better with enzalutamide. So this was a first clinical evidence to us that there might be some underlying molecular features that make those cancer cells less hormone dependent, more androgen receptor insensitive, and it's those same androgen receptor insensitive subsets that might in fact be more sensitive to the high-dose testosterone, whereas those patients who were responding extremely well to abiraterone for a long time, they probably benefited more from just going to another hormone blocker. So hold that thought for a while, because we're going to get to the biological underpinnings of that. But this was the part of the Transformer study that actually blew my mind and changed the way that I thought about this treatment. If you look at the red side of the curves there, these are patients that received bipolar androgen therapy, and then after they progressed, they got enzalutamide. Now keep in mind that if you get enzalutamide directly after abiraterone, the response rate is 25%. So what we seem to be doing here is we seem to be tripling the sensitivity to enzalutamide by giving an intervening therapy with the high-dose testosterone. So somehow we are rewiring those cancers, and we are turning back the clock from a more hormone-resistant lineage plastic form back into a more AR-addicted androgen receptor dependent form. And that was reflected also in a statistical trick, which is called the PFS2. I say that as a bit of a joke, but PFS2 is the combined progression-free survival through the crossover to the second progression after crossover. The red curve are patients that were randomized to BAT first and then crossed over to enzalutamide, whereas the blue or green curve are the patients that were randomized to enzalutamide and then crossed over to BAT. And here there was a statistically significant difference, favoring the BAT being given between, like a sandwich, between the abiraterone and the enzalutamide. So these two pieces of data made us quite convinced that in addition to the independent efficacy of bipolar androgen therapy, it also is doing a second thing. It's sensitizing to downstream antiandrogens. When you look at the PFS2 forest plot, you see the same association with the prior duration of abiraterone, where the benefit is greater in those patients with the shortest prior duration of abiraterone, again, suggesting that factors that might cause abiraterone resistance are the same as those that might cause BAT sensitivity. We also had the opportunity here in a randomized trial to look at a number of quality of life metrics. The red curves in all 6 of these graphs show the enzalutamide, the red curves show the BAT, and the blue curves show the enzalutamide. So the quality of life metrics are superior in all categories, including, to look at this carefully, physical function, energy, fatigue, sexual function, orgasmic function. So a lot of these sexual and orgasmic side effects were maintained and improved in patients who received the high-dose testosterone. So not only are we potentially giving these patients a benefit, not only are we potentially resensitizing them to downstream antiandrogens, but we're also improving their quality of life. And this is quite remarkable because in castration-resistant prostate cancer drug development, there really hasn't been another compound that improves quality of life at the same time as providing efficacy. So to prospectively test the hypothesis that alternating between high-dose testosterone and enzalutamide improves outcomes, we designed this study. And I'm proud to say that we co-designed this with Johns Hopkins, as Hopkins and Minnesota being the 2 lead sites. And this is a trial where we are taking patients with metastatic castration-resistant prostate cancer who have received abiraterone only, not enzalutamide and not chemotherapy. And then we're randomizing them in a 2-to-2-to-1 fashion. So they've got a 20% chance of getting standard enzalutamide continuously. We've got a 40% chance of alternating between testosterone and enzalutamide in 8-week cycles. So we call that the fixed alternating regimen. And then in the third arm, which is the other 40%, we have a variable alternating regimen, which is based on PSA. So in the third arm, we switch the drugs only when their PSA begins to go up by 25% or more. And the study has been powered to show whether either arm B or arm C is superior to arm A in terms of progression-free survival. So this is our 9th bipolar androgen therapy study, and in a moment I'll talk to you about our 10th. So we're making progress. Now, over the course of time, we had all observed a handful of patients that had achieved what we call exceptional responses to bipolar androgen therapy. And we define this very simplistically with clinical criteria. They had to have a minimum of a 70% PSA decline, a minimum of a 20% objective disease decline, and also a duration of response lasting a minimum of 6 months. And then we went back and looked at the next generation sequencing results from those patients. And what we found, and this was the first molecular insight, was that virtually every one of these patients in whom we had sequencing information, the white bars are the ones that we didn't have any DNA sequencing performed, either had a homologous recombination repair mutation or a pathogenic P53 mutation, or a combination of a HRR and a P53 mutation. And that was striking to us because it suggested that prostate cancers that have a deficiency in DNA repair, either homologous repair deficiency specifically or more global repair deficiency with P53 loss, might be the ones that are deriving more benefit. My fellow Michael Schweitzer, who moved to the University of Washington, Seattle, in conjunction with Pete Nelson there, did a study where they looked at homologous recombination deficient and proficient patients who received bipolar androgen therapy at their university. On the left, you can see that if a homologous recombination mutation was present, this was before the PARP inhibitor era, there was a 52% chance of success. Whereas if the homologous recombination status was proficient, only 18% of patients had a response. And interestingly, those 5 patients that responded on the right-hand side, 4 of the 5 had a pathogenic P53 mutation. So all the data appeared to be converging onto these 2 pathways, both of which are involved in DNA repair. And we then decided to ask an even bolder question. In the clinic, we know that there is a subset of prostate cancers that is defined histologically as neuroendocrine. Some people have used the word lineage plasticity, lineage plastic prostate cancers, which become more dependent on non-AR pathways. And in the most extreme form, these are morphologically small cell carcinomas. And at the molecular level, these cancers are characterized by combination tumor suppressor losses. And the tumor suppressor genes that are involved are P53, P10, and RB1. So we decided to do a study with 22 patients. It was hard to enroll these patients because they had to have 2 or 3 of the tumor suppressor. But we actually ended up over the course of about a year finding 22 of these patients. And we were absolutely amazed and struck that 46% of these patients, which have the most lethal variant of prostate cancer, were responding favorably to the bipolar androgen therapy. And while the median progression-free survival was not too striking at only about 5 months, what we showed was a very unanticipated, again, 22 patients only, improvement in overall survival, primarily due to the reason that these patients, again, were being resensitized to androgen receptor inhibitors, including ones that they had actually failed before. So we had patients with enzalutamide, they developed a tumor suppressor loss, they got bipolar androgen therapy, they went back on enzalutamide, and they had an 18-month response. So we were, again, sensitizing, perhaps reversing lineage plasticity towards an AR luminal phenotype away from a basal neuroendocrine phenotype. Interestingly, one of our fellows, Michael Hafner, he's a pathologist. He was doing his fellowship at the time at Johns Hopkins. He had made a very interesting discovery, which was that in LNCAP cells, which are BAT sensitive, if you treat those cells with supraphysiologic androgens, you can induce double-stranded DNA breaks as measured here by gamma H2AX immunofluorescence. And again, this is beginning to fit with the hypothesis whereby if you have an underlying DNA repair deficiency mutation or physiology, and then you induce double-strand DNA repair breaks, which are not able to be fixed by the cancer cell, it might be those patients that derive the greatest benefit. Now, after that, I made an accidental clinical observation that I was thinking about ignoring until it happened a second time. And the accidental clinical observation was this. We had a patient here who, if you look at the vertical line in the middle, received 10 cycles of bipolar androgen therapy after failing enzalutamide and Provenge. As you can see, he had a partial response. It did not last that long, maybe 7 or 8 months. He was in re-challenge with enzalutamide, as we were talking before, had a sensitivity, didn't last very long, went on to receive another cycle of BAT, got a chemotherapy, and then with a PSA approaching 60 and having exhausted all of his life-prolonging options, we were able to get compassionate use pembrolizumab. Why? For no other reason than he had nothing else remaining. There was no biological rationale. And within one dose of pembrolizumab, this patient's PSA went to 0 and remained at 0 for 18 months. Couldn't explain it. Now, if you look at his molecular sequencing, you can see 2 BRCA mutations. They were both categorized as VUSs, variants of unknown significance. He also had 2 pathogenic p53 mutations. So I wondered whether the bipolar androgen therapy was somehow immunologically priming his cancer for subsequent response to pembrolizumab. By the way, the response to pembrolizumab in prostate cancer is 2%. It's the only cancer for which pembrolizumab is not FDA approved. And the 2% of people that respond are those that have mismatch repair deficiency. And this patient clearly did not have that. He had a mismatch repair proficient cancer. And then it happened again. This time it happened with nivolumab. Again, compassionate use. This was a patient with a ATM pathogenic mutation, so this one is pathogenic. He also had an RB1 inactivation and androgen receptor amplification. This patient's amplification was like 25 copies. So that's a big time amplification. And again, this patient had received about 1.5 years of bipolar androgen therapy, then went through 2 chemotherapies, and then as a last resort, and almost like a Lazarus effect, we gave him nivolumab, which resulted in a complete remission for 22 months. And these were the scans of these 2 patients. You can see the first guy had a large retroperitoneal lymph node mass that was encasing his aorta, disappeared after 3 or 4 doses, and the other one had a less impressive, although robust response. So we decided to move beyond anecdotes into a prospective clinical trial, and this time we collaborated with Dr. Mark Markowski, who also had just finished his fellowship and had joined faculty and wanted to get involved in clinical trial design, and we designed the COMBAT study. So what we did here is we decided to test that hypothesis of whether the bipolar androgen therapy induces an immune stimulation. So we started with 3 cycles of high-dose testosterone alone. After 12 weeks of treatment, 3 doses, we performed imaging. If the patient had a stable disease or better, then he was allowed to continue on study, and we added on cycle 4 nivolumab, which was given on the same day as the bipolar androgen therapy. And then thereafter, every 4 weeks, the patients would get both high-dose testosterone and nivolumab on the same day. Again, the entire time we were maintaining their leuprolide and their medical castration. In this study, we were so intrigued by those biopsy results that we wanted to build in, and we actually made this mandatory. We made it mandatory that all patients have a metastatic biopsy at baseline. And then we had language that said, strongly encourage a second biopsy unless, deemed unsafe or infeasible. So we had a second biopsy 12 weeks after the first, and then we had circulating tumor DNA analyses. So let's review the clinical data and some of the biological insights. So we saw PSA responses and objective responses that were greater than we had seen with all of our BAT monotherapy studies, not 3 times higher, but the PSA response in all the prior studies was about 25%. Here we were approaching 45%. The objective response rate in prior studies was 10 to 12%. Here we were getting 24%. So at least a doubling of response. Now, when we submitted this to Nature Communications, actually went to Nature first and then it was bumped down. The reviewers wisely asked us, how can you tease out the independent contribution of the nivolumab? And so what we did in the figure on the right was we coded each bar in blue for the effect of the bipolar androgen therapy 3 rounds. And then in orange was the additional added value once the nivolumab was added. And you can see there's about 8 patients where their PSA decline became more deep when you added the nivolumab. There's also 1 patient shown on the right with the blue bar going up and the orange bar going down, where his best response was a PSA progression until we added the nivolumab, and then we totally reversed his course. He did not have a mismatch repair deficiency. So we are showing that there's a handful of patients, it's not the majority, but there's a handful of patients where we might be seeing some synergy between the two agents. Again, we seem to be getting consistent progression-free survival in the 6-month range, overall survival beyond 2 years. These were all patients, I forgot to mention, that had received 1 to 2 AR treatments and 1 to 2 chemotherapy. So these were patients that currently we would recommend the lutetium PSMA as the only FDA-approved treatment. In those days, they had no FDA-approved treatments remaining. Now, the safety of this was strikingly good. So it's rare that you see a phase 2 study with no or few grade 3 and grade 4 side effects. And the side effects here were mainly grade 1, grade 2. Musculoskeletal pain seems to be a class effect of the bipolar androgen therapy. We seem to be inducing small amount of musculoskeletal inflammation, especially in areas of arthritis rather than areas of bone metastases. We also see nausea in about a fifth of patients. Now, in this trial, we had one patient who developed immune pericarditis and cardiomyopathy. That was the same patient, which we think is due to the nivolumab. So it was 1 out of 45 patients, of about 2% risk, and that did not seem to be augmented. Now, here we had a very rich biomarker analysis. As I mentioned, this was a 45-patient study. 24 of the patients managed to have paired metastatic biopsies. You know, there's never been a trial before in metastatic prostate cancer with paired biopsies on this many patients. So the first thing is that the effect on androgen receptor protein going down was not a one-time artifact. It was absolutely consistent. So in every patient, whether they responded to the therapy or not, this occurred also in the non-responders. At the protein level, the quantitative immunohistochemistry of AR went down. And if you look at the far right, the blue dots are the responding patients, the red or pink dots are the non-responding patients. You can see in general, although there was not a statistical trend, that there are more blue dots at the bottom and red dots at the top. But again, there was not a strict correlation between the degree of androgen receptor protein reduction and efficacy. We also looked at Ki-67 again at the protein level. And here we didn't see a uniform pattern. We saw some patients, like the one shown on the left, where the Ki-67 eventually disappeared. This was also a patient who had an objective response, but there were clearly other patients where their Ki-67 either did not change or went up. Again, here, the statistics were not significant, but the ones that had the Ki-67 reduction were more closely associated with response. And then the most intriguing thing to our pathologist, Angelo DiMarzo, who's a MYC aficionado, is that in some cases there was a profound downregulation of MYC. And MYC has been an elusive target in prostate and other cancers, lymphomas, for a very long time. And again, this is at the protein level. And here there was statistical significance. The patients that had a BAT-induced MYC downregulation at the protein level had a significant improvement in their outcomes. And this is shown here in a separate abstract that we focused mainly on the MYC. First of all, MYC and Ki-67 correlate with each other. The patients that have higher Ki-67 also had higher MYC, and lower Ki-67 had lower MYC. And the Kaplan-Meier curve shows that after receiving bipolar androgen therapy, if the MYC level at the protein declined by 50% or more, those patients had a longer, I think that's progression-free survival. Yeah. The next thing that we did is we took the baseline biopsies. And again, we had 24 paired biopsies, but we had about 40 biopsies where we had the baseline only. And we did RNA sequencing whole transcriptome analysis of the baseline samples only from these 45 patients. And we ran gene set enrichment analysis on that to ask the question, what transcriptional programs at baseline before the first dose is given predict a favorable response? And there were 5 things that met statistical significance. The first was upregulated androgen response signaling. The second was impaired DNA repair. The third was accelerated E2F signaling. The fourth was upregulated MYC. And the fifth, which we did not expect, was upregulated Wnt beta-catenin. And the strongest correlation was with upregulated AR, not just the AR gene itself, but the AR transcriptional program, at least the one that's included in the hallmark gene set. The next question was a pharmacodynamic question. What type of changes to the transcriptome are induced by the bipolar androgen therapy? And this volcano plot answers that. Everything that is on the right-hand side shows pathways that were increased as a consequence of BAT, and the left shows pathways that were decreased or downregulated as a consequence of BAT. The red dots are the ones that distinguish the responding patients from the non-responding patients. So the pathway that was the most upregulated and correlated with response was this allograft rejection immune signature. There was also a complement immune signature. There was also an IL-2 immune signature. So those three were the ones that were the most induced and associated with response. And then the ones that were most downregulated and associated with response were MYC and E2F and G2M cycle checkpoint. We then looked at the baseline biopsies from these patients to do a DNA-based whole genome sequencing. We're still going through terabytes of data on this, but this is the first pass where we looked for just pathogenic alterations as defined by the OncoKB website. And what I'm showing here, and all these confidence intervals are very, very wide, and keep that in mind, but we're just looking for trends here. The 43% is the PSA response for the entire population. So anything to the right of that are genetic signatures where the mutation induces a better response than the average. Then the ones in red are the genes that if those are mutated, patients have a lesser response. So the highest ones inducing a favorable response, once again, the pattern keeps coming up again and again. HRR mutations and P53 mutations. And interestingly, for those of you in the prostate cancer field, SPOP mutations, which are present in about 10%, were associated with resistance to this treatment. We were intrigued by the P53 finding, and I reached out to Michael Schweitzer, who's now an associate professor. He's growing up so nicely. And Michael had done a trial at the University of Washington, Fred Hutch, looking at the combination of bipolar androgen therapy and a PARP inhibitor. And his hypothesis had been that the patients that have homologous recombination mutations would do better. And in fact, the trial did not show that. There was no difference in bipolar androgen response in his prospective study in that combination, which I found to be surprising and unexpected. However, what he did find a difference was in the subset of patients, which was about 30% of the total, who had a P53 mutation, the combination of BAT plus olaparib produced a statistically and clinically significant improvement in median progression-free survival. So that was really interesting and led us to focus on P53. Now, Angelo DiMarzo was able to do a 8-color immunohistochemistry for a number of immune cell subsets from this trial. And this is an example here showing CD3 stain in that purple color, CD8 stain in yellow. And then you can fuse the two images where you can look at the CD3 positive, CD8 positive cells shown in orange. Then you can overlay the PD1. So you can ask yourself, what's the proportion of PD1 positive, CD8 positive, CD3 positive cells? And when we did that, from the baseline samples, this is not a pharmacodynamic marker, this is a baseline marker. We found that a number of cell subsets at baseline were associated with response to the BAT-nivolumab combination. For example, PD1 positive CD8 cells were enriched in the responders, PD1 positive CD4 cells, and then the total number of PD1 positive CD4s and CD8s. So this led us to believe that if there was a tumor microenvironment at baseline, which enhanced inflammation, at least a PD1 positive CD8 and CD4 inflammation, this could be a marker of response. Now, I want to spend the last 10 minutes talking about an effort that some of you may have heard about, which is a joint effort with the Mayo Clinic to try to submit a prostate cancer spore grant, a specialized program in research excellence. This is a clinical grant whereby you have a minimum of 3 projects, supporting cores, and every project has to have a human clinical endpoint, either a clinical trial or biomarker assessment. And this figure is a bit hard to read, but we're going to be submitting this in May, so we're 2 months away from crunch time. And Scott and I will be leading that effort from our side. And Dr. Oliver Sartor, who some of you may know, and Giuseppe Domingo Dominic will be leading it from their side. So we have 4 projects in this grant proposal, 2 from our side, 2 from the Mayo Clinic. And Scott and I are leading the project 1, which of course is related to bipolar androgen therapy. I was intrigued by the fact that P53 mutant prostate cancers have an abysmal prognosis. We have no therapies directed specifically at those patients. It's easy to detect the mutation status because all NGS platforms do it, and it can also be detected routinely from circulating tumor DNA. And the hypothesis for this trial is that if we combine bipolar androgen therapy with another DNA damaging agent, in this case carboplatin, which is a generic platinum therapy, we would induce synergy. And we have 3 aims. The first aim is the clinical trial. The second aim is to use the biopsy samples from the trial to understand biology. And the third aim is going to be done in Dr. Dame's lab to understand preclinically what's going on. And these figures were created by Scott, and I think they were eye-opening. And these are novel insights that the field did not previously have, and they're not published yet. There's a very strong link between androgen receptor alterations, especially amplifications, and the presence of P53 alterations. So on the left-hand side, Scott used a circulating tumor cell data set from an Alliance trial that Chuck Ryan was involved with. And on the right-hand side, he used publicly available West Coast and East Coast stand up to cancer tumor biopsies. And the conclusion in both data sets was the same, that patients that have pathogenic P53 mutations also have an increased amount of amplified AR and also structurally rearranged AR, so genomic structural rearrangements, including a very unique type of rearrangement, which is called extrachromosomal circular DNA. These are extrachromosomal DNA fragments. They used to be called double minutes back in the day. They're now called ecDNA, extrachromosomal DNA. And Scott's lab discovered that there's a unique translocation between the androgen receptor gene on chromosome X and a gene on chromosome 16, where you have a circular molecule that is derived from this translocation. And that circular molecule can be propagated from cell to cell because it's in the nucleus and then is extrachromosomal. So here's the first aim. It's a randomized trial. We're going to be comparing a combination chemotherapy, which is considered standard in these patients, cabazitaxel plus carboplatin, against the combination of high-dose testosterone plus carboplatin. This study is going to have mandatory biopsies at baseline and again at week 12 if it's safe and feasible. I'm going to skip some of these highlights about the trial. The second aim is going to use DNA sequencing, RNA sequencing, and also fluorescence in situ hybridization to look for these extrachromosomal DNA molecules. And Scott's lab has perfected an assay that he's now tested on not just cell lines, but also in human biopsy specimens. And there's a fluorescent signal that is given when the fusion of that X chromosome and chromosome 16 is overlaid. And we're going to be looking also at the RNA level at specific gene signatures that might be responsible for the efficacy. Interestingly enough, the canonical androgen receptor gene signature does not appear to be as important, but there's a second androgen receptor signature, which is called the proliferative or non-canonical or androgen variant mediated signature that does appear to be at least prognostic and maybe predictive. We also found using data that was recharacterized by Scott's group that there are downregulations of DNA replication and HRR repair in patients, both in the responding and non-responding group. But if you look at the non-homologous end joining and the Fanconi anemia pathways, those two are pathways whereby the responding patients have a greater degree of downregulation compared to the non-responding patients. So there might be some types of DNA repair deficiencies that are more prognostic of response than others. In aim 3, which is going to be a preclinical aim, the Scott Dame lab is going to look at a number of existing BAT sensitive and BAT insensitive cell lines and patient-derived xenografts, some of which have P53 deficiency, some of which are P53 proficient. And he will look at the combination of BAT alone, carboplatin alone, and both. And I want to focus your attention on a very interesting figure that I received yesterday from Scott, literally yesterday. And this is a patient-derived xenograft model with a LUCAP CR cell line, which is one that we've known for a while and Scott has confirmed is sensitive to bats. So these are 5 mice or 6 mice. I don't remember, Scott, if it's 6 or 5. During the green part of that curve, after tumors had grown to a certain size, they were exposed to serial supraphysiologic testosterone injections. The mice were then propagated until they developed a secondary resistance. And then what Scott's lab did is he compared the tissues of the bat-sensitive phase, which is the LUCAP-CR, to a second phase, which he called LUCAP-CR BAT-R, which means bat resistant. And the first thing that he found is at baseline, this cell line has an enormous number of these extra chromosomal AR circular DNAs, as you can see in the very hot pink fluorescent staining on the left-hand side. And amazingly, when these cancers develop resistance, instead of further amplifying the AR as they would if this was enzalutamide or an anti-androgen, they do the exact opposite. They downregulate these extra chromosomal amplified ARs. So this is fascinating, and it suggests that the amplification of AR, especially in these extra chromosomal circular DNAs, might be one mechanism or potentially the driving mechanism of sensitivity. And we'll be testing this also in our biopsy samples from the trial. So why is this innovative? At least in my opinion, this is the first trial to specifically target the P53 mutant subset. By the way, it's about 30 to 35%. Prostate cancers is not rare. The prevalence goes up in the metastatic CRPC setting where it can be as high as 50%. It capitalizes on a decade of understanding and more than 400 patients in 8 trials. We're going to be using metastatic and circulating tumor DNA biopsies and co-clinical trials in patient-derived xenografts. So we're trying to turn history upside down. We started with Huggins and Shalley, and we're going to do the exact opposite. We're not listening to our forefathers, and we are actually trying to go against the grain, break some icons, and create some paradoxes, hopefully for the benefit of patients. And we're going to look and take a deep dive prospectively now in a lot of these biomarkers, including this novel concept of androgen receptor extrachromosomal DNA. And I'd like to finish with that and remind everyone, including myself, that we've never had a treatment before that improves quality of life rather than, the conversation we always have is, this is the benefit and these are the side effects. And now we're saying this could be the benefit and these are the things that you might feel better with. So it's actually an interesting conversation to have with patients and very rewarding. I'd like to thank Scott for all his collaboration and friendship. Thank all of you. And that's all I have. There's going to be a microphone for questions, so raise your hand and grab the mic.
All right, lots of questions.
So my question is, do you think the AR-dependent CRPC is more sensitive to the BAT than the AR-independent CRPC, like NEPC or something?
The androgen receptor has to be present in order for this to work. The androgen binds to the androgen receptor. So if androgen receptor is completely lost, like in pure small cell prostate cancer, where at the protein level, AR is simply not there, this won't work at all. But to get back to your point, there's an entire lineage between a pure AR positive prostate cancer and a pure small cell cancer, where some of the genomic hallmarks, such as the P53, RB loss, PTEN loss, where those cancers are being driven closer to an AR-independent phenotype, but they're still expressing AR. And I think in those patients, we have an opportunity to reverse the lineage plasticity, but if AR is lost completely, this won't work.
Daniel, that was terrific. So just a comment, when I was in medical school, Dr. Huggins still had a lab, and he was painting lipstick on mice because he believed some of the dyes actually had steroid hormone properties, which I think he was right about too, because they do. But I've always think it's really interesting that both breast and prostate have similar observations, but they then go in different directions with those observations. So question number one, and this may be for Scott or both of you. In estrogen-deprived breast cancer, a decent number of patients have mutations in the hormone-binding domain and AF2 domain. So the response to that in the breast cancer world has to build estrogen receptor binding drugs that target that phenomena. And the second observation, at least in breast cancer, is okay, so they're estrogen independent or sort of estrogen dependent. Let's hit other pathways like PI3 kinase pathway or CDK4/6. But you guys are a little different, I think, in terms of how you're thinking about this similar problem.
We are similar and different. So we do have AR ligand binding domain activating mutations. Those tend to respond less well to antiandrogens. We don't have AF1 or AF2 domain mutations. So we don't have any N-terminal domain mutations. There are some new strategies to reduce the number of promiscuous ligands, such as other steroid receptors that can bind to those mutations. They haven't worked extremely well. Other pathways like CDK4/6 have been a bust in prostate cancer. And even the PI3 kinase pathway in those that have known PTEN deletion, which can be measured routinely now with immunohistochemistry, even those studies have produced very, very modest PFS benefits. You know, you can hardly see it, no OS benefit, and we have no FDA approvals.
So, give the microphone.
He doesn't have a microphone.
I didn't see him behind.
Yeah,
Dr.
so
Simon.
Emmanuel, that was amazing. It's such a tour de force, and there are so many questions, but I'm going to limit it to two that I hope you can answer. One is side effects. Do you see any changes, increase in behavioral aggression? Biology, I was really intrigued by the immune response. And, you know, you showed us one example of a met lymph node where the tumor had gone down. It's really hard to see in an IHC, but it looked like the rest of the node was pretty trashed. So I wonder if you've looked at all at how the immune environment in the tumor and the lymph nodes is changing, that's sort of priming that immune response, and whether there's any relationship between the ability to reform follicles in the node or having tertiary follicles in the tumor is actually going to be an indicator of response.
So side effect profile, yes, there is some increased aggression. Sometimes it's mild, and sometimes it's reported by the spouse rather than by the patient. So I'll just give you a real example. One of my patients, you know, his job was always to wash the dishes and take the trash out. And he went on the study, he just started to refuse that. He said, I'm not doing that. I'm not going to take the trash out. There wasn't like an overt aggression or physical aggression. The other thing is nightmares, not just dreams, but nightmares. And there was one patient that repeatedly was seeing the same dream, that his wife was trying to stab him in bed while he was sleeping, and he would wake up and he would swipe over at her and she would have to move out of the way. So yes, there are some psychological, psychiatric, those are the minority of patients, and those are really hard to capture with the classical, you know, CTCAE side effect lists. You know, the immune analyses, we're just scratching the surface. We didn't have the foresight or the insight to capture the biopsies in a fresh manner, so we can do single cell analysis. So we can't do single cell analysis, so we can just do the bulk analysis either by immunohistochemistry or immunofluorescent methods. And at the first phase, we were just focusing on quantifying the intratumoral immune milieu. So Angelo DiMarzo was circling the malignant glands and excluding any cells that were in the stroma. So everything that I showed you is intratumoral. What is interesting, and we've known this for a long time, is in prostate cancer biopsies and primaries, 80% of the immune cells are in the stroma or in the stroma tumor interface, and the tumor itself is a lymphocyte desert. And so it was encouraging that even within the tumor, we were seeing increased subsets, but we have not really studied yet the interaction. And I think that's an interesting next step.
Emmanuel, great talk, amazing talk. Two questions. One is actually an observation. In your Nivo and Pembro responders, it looks like you're kind of borderline TMB high tumors, right? So it could be that it could be that it's not. I mean, TMB high prostate cancers don't have amazing responses to immunotherapy all the time, but you could be augmenting that process somehow. So that's a potential confounder. But the other thing that I am always struck with when I see these pre and post treatment biopsies is you say, okay, well, we lost the AR, But really what you're looking at in those post-treatment biopsies is you're looking at the cancers that the tumor cells that didn't die. And so what you're really looking at is potentially your future resistant cells. And so it's one thing to say, well, we're down regulating AR, but it's another thing to say these persistent tumor cells harbor this and that feature. And so I guess what I'm asking is, in those residual tumors, are you seeing evidence that what you're doing is simply selecting for potentially lineage plastic type cells?
Yeah, we haven't gotten there yet. We have this deep treasure trove of the whole genome sequencing, and we're working with Vasan Yegna-Subramanian, who you may know, on that. One of the things that we're trying to confirm in that study is whether these double-strand breaks and rearrangements are augmented at week 12 versus week 0, and also whether the Tempers-Erg fusion, which is the most common fusion of prostate cancer, whether we can induce that in some of these patients. So we don't have that analysis yet. We've done the RNA data comparing week 12 to week 0, but we have not done the DNA-based analysis yet. But I like your point, and in many of the neoadjuvant pre-prostatectomy studies, the tumor left behind at the time of prostatectomy can teach us about the emerging resistance mechanism. So maybe instead of viewing this as a pharmacodynamic measure of benefit, I should be focusing on what's left behind.
That was what I did as a fellow. Looked at those residuals.
All right, we're out of time, so let's say thank you one more time for a great talk.